Well, it is really a pleasure to be here. So indeed, I did a very brief postdoc in the Eversold lab in 2005. So I was just saying that it's kind of feels like coming back home every time I visit. It's really nice to be here. And so I'm a statistician by training. I work in all things quantitative mass spectrometry and proteomics. And so for the two lectures today and tomorrow, I would like to tell you about the experimental design and then basic statistical analysis and also some more advanced methods that we use specifically to analyze targeted quantitative proteomic experiments. So first of all, why statistics? So as we kind of probably have a sense already, it's important, but why, right? So what exactly we're trying to do with statistical methods? And well, the point is that in quantitative experiments, variation and uncertainty are unavoidable. And there are many types of variation and uncertainty that we have to deal with. So from the experimental perspective, when we are analyzing the components of a biological sample, so we worry a lot about technical variation. So sample handling, storage, processing, how much different types of sample handling affect what actually comes out as far as the data at the end. There's also instrumental variation. Elution time, ion suppression, all of these things can affect the peaks and the quantitative readouts that we have from these peaks. Computational signal processing, peak boundaries, identity, intensity of the peaks, all of these things can also be either variable or not quite clear how to find the peak, how to integrate peak. And so these are kind of all the technical things. And sometimes I hear from my experimental collaborators, you know, if our experimental workflow is optimized enough, if our instruments are accurate enough, and if my whole procedure is precise enough, then I don't need statistics. And this is fundamentally wrong, because even though we can do our best and do a really good job to take into consideration all of these things, what we still have left is the biological variation. So even if our measurement system is perfect and infinitely accurate, as soon as we deal with biological systems, we have a natural biological variation of the amounts of proteins that we have between one individual and another. And even if I'm not talking about humans, even if these are cell lines or yeast samples, there is a lot of biological variation that we have to deal with. And so statistics can be thought of as a body of methods for making wise decisions in face of uncertainty. And so as soon as we have biological variation, we have uncertainty. So we need to know what are the systematic signals which appear, for example, if you induce stresses or some kind of treatments to the samples versus what is just the random variation. And so contributions of statistics actually come in three ways. So one is the experimental design, and that's what we will talk about today. So how can we use statistical considerations to design the most effective experiments? Then statistical analysis. Once the data are collected, so how can we integrate all the available information and take it to some kind of objective conclusions which are not relying on the subjective opinion of a particular person who does the analysis. And the last part is statistical software, because even if we design experiments and if we can analyze this, the important aspect of this is reproducibility. And so if we can repeat the analysis using statistical software, and if we can have an objective procedure which provides the conclusions, then somebody else can reanalyze the data, but more importantly, somebody else can repeat the entire experiment and repeat the entire analysis workflow. And this is really important. So what I will do first, before I jump into the experimental design, I will talk a little bit about how we can translate the scientific question into the statistical question. So oftentimes when my collaborators come, they come with questions such as, well, no, I have a biomarker study, so how can we approach this problem, right? And so what goes on in my mind is I'm trying to translate the clinical or the biological question into a very specific statistical framework. And so that's what I will tell you first. So what are the types of analysis in a very broad sense, right, that we can think of when we talk about a particular experiment. And just for these 2 days, I will talk about biomarker discovery just because it's kind of simple to think about. But it doesn't have to be biomarker. It can be any basic biology investigation, systems biology studies, or whatever you have. But I will just talk about biomarkers to be somewhat specific. And then we will talk about these things. Okay? So 3 types of statistical goals. So when you say I have some kind of study, I have a, let's say, biomarker discovery experiment, that's what goes on in my mind. So the first statistical goal is class discovery. So, and we will look at things such as like that. So for example, here we will have different proteins, here we'll have different samples, and the color corresponds to the abundance of the proteins on some kind of relative scale. And we want to say, well, I want to find proteins which behave similarly across samples, or I want to find samples which are similar somehow in the space of proteins. So both are valid, right? So for example, if you're working on a clinical experiment with biomarker discovery, maybe you have some broad definitions of disease, like healthy and disease, and maybe you're looking for subclasses which are more homogeneous in the kind of space of protein abundances than just disease, because maybe there are some subgroups of the disease. And the same for the proteins. Maybe you look for homogeneous protein groups which behave somehow similarly across all the conditions. And maybe it's important because maybe these proteins are somehow involved in the same mechanism. So the point here is that we do not know what we're looking for. We have no class labels given in advance, for example, no kind of definitions of the groups. All the variation is treated equally. We say, well, we just want to look for patterns which are consistent across proteins or across subjects. And so we are looking for something, we don't know what. And so it is often difficult to find some meaningful answers if we're not telling to the statistical method what exactly we're looking for. And so we look for some patterns of similar variation, and it may be systematic variation, it may be noise variation. We don't know that because the analysis does not know what we're looking for. And so from this perspective, it's called an unsupervised method. Unsupervised means that we did not give any labels to this method in advance. And so the common mistake that people make, so for example, they find some kind of pattern, so this will be groups of proteins that behave kind of similarly across samples, or this would be principal component analysis, where each point here is a sample, and the coordinates are some kind of combinations of the proteins. And essentially, if people find these patterns, they say, oh, look, there are some kind of changes, therefore we find biomarkers of disease. This is wrong. Okay, so the unsupervised methods where we don't know what we look for cannot lead to discovery of biomarkers. And this is because we do not give a definition of what we're trying to find, so we don't have a definition of the disease type or class, and also because we don't give the group labels, we cannot report error rates, we cannot characterize the performance of these methods. So these unsupervised class discovery methods, they are only good for data visualization or for initial summarization of things. And so these plots are very much useful, but the point is that this is not the end, this is just the beginning. So we cannot just stop here, and if we have some kind of nice colorful patterns, it does not mean that we have biomarkers of disease. We could just represent this multivariate data set in some kind of visual way that we can kind of start looking at the data. So that's class discovery. So discovery means we just start from something, we don't know what we're looking for. So the second statistical goal is class comparison. So now this is a supervised method in the sense that we have the information on the samples. Okay, these are subjects which are healthy. These are subjects which come from the disease group. And so now we want to know if there is a change in protein abundance between healthy and disease, on average between the groups, more systematically than as expected by random chance. Okay, so that's what it would look like. We have a healthy subject, healthy subjects, we have disease subjects. Each dot is the protein abundance in the group. The line is the mean of the protein abundance. Maybe this is on some kind of log scale. And so the question is this change in the mean more systematic than as expected by random chance, given the variability around the mean? So this is already interesting because now we're distinguishing the systematic variation from the random variation. Okay, so we can compare group averages. Typical methods will be t-test, ANOVA, some kind of permutation tests, if you have heard about them. So the readouts of these methods will be p-values or posterior probabilities, depending on the methods. The important thing is that we compare averages between the groups, right? So we say, well, the average over these healthy subjects is different from the average of these disease subjects, okay? And so if we have a small p-value, it means the difference is more systematic than as expected by random chance. The point is that proteins with small p-values are not biomarkers. And this is, again, a very common mistake that I see, that when people do some kind of t-test and they have a small p-value, they say, we found a biomarker. This is not true, because a definition of biomarker is something which is actionable in clinic. So a doctor sees a patient and needs to decide if this patient has a disease or not. The doctor does not see the average patient, right? Here we compare averages. The doctor needs to know what is going on for this particular person. And it's okay, it doesn't matter if on average over many, many subjects the protein changes. The question is what is going on with this particular patient. So here we compare the population averages. This is very useful. And oftentimes if there's an average change, it is possible that for individual person there will also be some kind of change. But this is still not enough to claim the biomarker. So this is good for initial screening experiments. This is good for basic biology studies, but this is not enough for discovery of biomarkers. So this is just a couple of slides to emphasize this a little bit. So this is one example where proteins with small p-values are not necessarily biomarkers, so they're not necessarily predictive. So let's take a look. So we have two groups, healthy and disease. So each dot will be the protein abundance in each sample. So the line will be the mean in each group. So here we see that the change between the means, well, it's fairly large as compared to the variation around the means. So if a person comes from, with, let's say, this protein abundance, it's very likely that this person comes from the healthy group. Okay, so in this case, it's differentially abundant and predictive of the disease. But here's another situation. We have a very large sample size. We have a large study, so there's a lot of variation around the mean. So it is possible that this change is an artifact of random chance. But now we have so many subjects that we're really, really confident in this mean, and really, really confident in this mean. So this situation will typically give us a small p-value, because this change in means will probably be viewed as a systematic change. However, if a person comes with this value of protein abundance, is this person coming from the healthy group or from the disease group? You don't know. So even though on average the change is systematic, it is not predictive of the disease status of the individual patient. And so this is the case where a protein with small p-value will not be a good biomarker. And so if we're really looking for biomarkers, we need to clarify that we're really looking for predictive models and not just for looking for changes on average in the population. And here's another situation. So now this is the opposite, where we can have a biomarker which is predictive, but it doesn't necessarily have a small p-value. It doesn't necessarily change between the groups. So here is a situation where we have the blue dots will be healthy subjects, the red dots will be disease. We have two proteins. And so you see that the protein 1 is low for healthy and high for disease. Protein 2 is low for healthy and high for disease. So this will be both differentially abundant and predictive. But let's take a look at this situation. So the healthy subjects span the entire range of abundance for protein 1, but so do the disease subjects. Right? And here also, the healthy subjects span the entire range of protein 2 and also the disease subjects. But as a multivariate panel of two proteins, a combination of abundances of protein 1 and protein 2, they're perfectly predictive. Right? So low protein 1 and protein 2 means healthy or high, right? And then the opposite means disease. This is, of course, a cartoon, right? So in real life, it will not be like that. But this is a case where you can actually use a multivariate panel of proteins where the proteins are not necessarily differentially abundant. So the point is that when we start, well, okay, so let's maybe finish here. So the third statistical goal, so this will be something that we need to pursue when we're really interested in class prediction, is actually trying to predict the disease status of the individual subject. So now again, it's a supervised method. We have known class labels. But now our goal will be, let's say if we have two proteins, is to find some kind of decision rule which classifies the individual subjects into healthy and disease. So here I have blue will be healthy, red are the disease. Here I have a simple rule where I say protein 1 doesn't matter. Low values of protein 2 mean disease. High values of protein 2 mean healthy. Okay. But we see that there are some red dots kind of appearing here and blue dots appear here. Maybe this rule is not very good. Maybe you can have a more complicated rule, which involves both protein 1 and protein 2, which does a better job. It classifies healthy more systematically and disease more systematically. But hey, we can have an even more complicated rule. We can have some kind of really fancy line here, which distinguishes red and blue dots perfectly. And it's possible that this rule is actually something reasonable, which will be reproduced in a future study, but likely this rule kind of adjusts too much to the specific subjects that we have in our study, so probably it is too detailed that it will not be reproduced in the future study. So this is an example of overfitting, where we adjust too much to this particular data set. So this is too simple, this is too complicated, this is just enough. And so the goal of a statistical analysis would be to find a rule which is kind of like a Goldilocks, you know, not too little, not too much, but just enough to do well in a future experiment. And so the type of readouts that you will have is misclassification rates, sensitivity, specificity, predictive value. And so this is something that clinicians are very familiar with, and they kind of think in these terms. And so this would be useful for when you are moving beyond the initial screening experiment and look for more kind of confirmatory studies. So the point is that before we start the study, we need to even decide first what type of experiment this is and what class of statistical methods we are thinking about. So are we talking about class discovery, where we have no particular labels in mind, we just want to look for patterns? Are we in the initial screening experiment, where we just want to compare the averages between subjects, and we will use this, maybe in the next iteration we will do a larger study? Or are we already in the kind of confirmatory stages of the experiment, where we try to now increase the sample size and actually go down to the specific kind of predictive models which can potentially be used in clinic? And so this would mean different statistical methods, and this would mean actually different experimental designs for that. Let me take a pause here. How does it look? Do you have any questions? I'm really happy to answer questions, because I can talk for a long time. It's all right? Okay, don't hesitate, okay? Interrupt me anytime. So now what I will do, I will focus on this second part. So I will focus on this goal where we'll look for comparisons between the means of the two populations. And we'll talk about the dangers which can appear when we are working on this type of experiments, and also how to prevent some of the undesirable situations. And so this is, well, for targeted proteomic experiments, this is the type of statistical goal that occurs very frequently, right? So we're not exactly just discovering some kind of unsupervised patterns, but we're not quite yet, or rarely we're in a very large scale experiment. So usually it is some kind of intermediate stage. Okay, so let's talk about the experimental design for a little bit. So just as at the very beginning, we defined what is the type of the statistical analysis that we're looking into. So once we agree that we talk about class comparison, so now we need to think about more details of how exactly we are going to proceed with the analysis. And so when our collaborators come to me and saying, well, now that I know that I want to do class comparison, so how can I design the experiment in the best possible way? Well, again, there are some abstractions which come to my mind, and I wanted to share this with you, because if you know how statisticians think, it can help you also communicate with them, right? And so let's imagine that we have two groups. So we have a healthy population and a disease population. Well, let's say healthy and cancer or something like that. And as far as all the risk factors or other types of confounding factors which can affect protein abundance, such as, let's say, other diseases, gender, ethnicity, weight, smoking, you know, whatever you can think about. So these two populations are identical, except for the fact that these subjects are healthy and these subjects have the disease. But they all have other factors which are very similar. So we have as many, let's say, male and female, as many younger and older subjects in this population. So we compare populations which are very similar, except for the fact that they differ in the status of the disease. Now, these populations are very large, right? So usually this will be, I don't know, maybe all the Caucasian subjects, all the European subjects, and so on. So of course, we cannot quantify protein abundance in these subjects exhaustively, right? So what do? We select a random sample from these populations, and we include it in our study. So this gives us the first layer of variation, because, as we said in the beginning, there is a natural variation protein abundance from one person to another. So if we wanted to repeat the experiment and do it again, then we'd have different subjects in the study, and the subjects will have a different natural abundance of proteins, right? So if we are to repeat this, we will not have exactly the same people, so we will not have exactly the same protein abundance. But that's not the whole story, because now we still don't know what is the protein abundance of the subjects. We measure this abundance using a measurement technology which is itself noisy. So the only thing that we see is this, right? So these will be the noisy measurements on the selected individuals, and so we have two layers of variation. One is the biological variation due to the fact that we selected the individuals randomly in the study, and the other is technical variation, which is due to the fact that our measurement workflows are not exactly kind of repeatable, right? So we have some noise and uncertainty and variation associated with that. And so now we have, because we have two layers of variation and uncertainty, we have two types of dangers. One is the bias, and this is really the worst type of danger. So bias means that the conclusions based on what we observe are systematically different from the truth that we're interested in, which is what happens in the actual populations, right? So this is what we're interested in, this is what we do, this is what we observe. Now we try to use the observed data to make conclusions about what we don't see, about the underlying populations. And this, by the way, is what is called statistical inference. We use the data to infer something about what we don't see. So this is what happens in these larger populations. And so bias would mean that we systematically do something wrong. So for example, if we have all the healthy populations Caucasian and all the disease subjects African American, so then the difference in protein abundance can be due to two things, right? So it can be due to the disease, but also to the ethnicity. And it's possible that a particular protein is affected more by the ethnicity than by the disease. We don't know this, so we find a difference. We say, hey, there's a difference. We attribute this to the disease when it's not the case. We repeat the experiment, we do the same thing, we see it again. So bias is reproducible. And this is why it is so dangerous, because if you try to repeat the experiment and you make the same mistake, you will see exactly the same wrong result, and you will not be able to realize that there is something wrong with that. So this will be one example, but even from the technical kind of aspect, so if we make some systematic mistakes somewhere in the process of data collection, again, we will be able to reproduce the same differences which are not due to the disease. So this is really the most dangerous thing. But then there's also inefficiency. So if our whole process has a lot of variation, so we have a lot of biological variation and a lot of technical variation, then the data that we use are too noisy to really make any reasonable conclusions about what's going on in the underlying populations. And so if we have too much noise, then it's not an efficient experiment, because we just cannot really tell much from the noisy data about what's going on in the underlying populations. And so the goal of statistical experimental design is to eliminate bias and to eliminate inefficiency. And this is something we do before collecting the data. So this is really important. So if we worry about these things after we collected the data, so there's really not much we can do. So there's a famous quote that asking a statistician to kind of work with the data after it was collected, it's like asking the doctor to look at a diseased patient. So at best, the doctor can diagnose what the patient died from, but you cannot really resuscitate that. And so it's the same thing. So at the best, we can say what went wrong in the experiment. But if an experiment was designed in a way that it had bias or inefficiency, then no amount of statistical sophistication will save that. And this is a really important point. So if there is one message I really want to communicate to you, is talk to a statistician before collecting the data, before even starting. This is really, really important. So Well, now I won't do a whole lot of math, but let me just kind of do one more step to show you what is actually going on in my mind when we discuss this type of things. So there is some kind of constant, so this will be the mean abundance of the protein among the healthy subjects, the mean abundance of the protein among the disease subjects. Now, each selected individual has its own protein abundance. What we really observe is the peak intensities, and then we try to look at the differences between peak intensities, typically on the log scale, to make conclusions about the differences of the abundances of the proteins in the populations. And bias means that this difference is systematically kind of deviates from this difference. And inefficiency means that the variability associated with this data is too large. So that's really what's going on. And so statisticians will formalize this with kind of more kind of specific quantitative models. Okay, but before we get into that, so how we actually prevent bias and inefficiency? And we prevent this with 3 fundamental principles of experimental design, which have been around for over a century. And some of them are very simple and others can be increasingly sophisticated. So the first fundamental principle is replication. Essentially, we need to take replicates, right? And so I'm hoping that by now there's enough understanding in the community that it is important. However, I'm still reviewing papers with sample size 1. You know, it still happens. So why replication is important? For 2 things. So one is it allows us to carry out the inference. It actually allows us to go from here to here, from the data to kind of quantifying what is going on in the underlying populations. And also, more replicates means more signal, right, which can cut through the noise, so it minimizes inefficiencies. So 2 purposes, right? So let's take a look. So let's imagine just a very simple experiment. So even though we talk mostly about SRM and DIA, just kind of for the moment, let's think about a simple LCMS experiment. So we have proteins digested into peptides. We have 1 peak for the protein for the healthy, 1 peak for the protein for the disease. So our question is there a systematic difference between the healthy and the disease? So if we have no replicates, well, we have 1 value for the healthy, 1 for the disease. Is it systematic or is it random? Who knows, right? We really don't know. There's no way to tell, and that's kind of the point, right? So if we have only 1 replicate, we see a difference, but we don't know if it can be an artifact. Now, if we have replicates, in this case, we see that the variation is small as compared to the change in the mean. So this is not likely to be an artifact. This is likely to be systematic. On the other hand, if the variation is large, exactly the same change is more likely to be an artifact, right? So just kind of to remind you why I'm always thinking in the log space. So so the difference of log intensities is log of the ratio, right? So when I talk about the difference here, it's the same as log ratio between the groups, right? So this is, and this will be the same log fold change or the same fold change, which may or may not be systematic, depending on the underlying variation. So using fold change alone as a cutoff is wrong, because the same fold change may or may not be an artifact of random variation. So fold change alone is not enough. Okay, and so this kind of illustrates that. So now we need to have replicates, and we need to do more than just look for the fold change. But remember, we have 2 layers of variation. We have the biological variation and we have the technical variation. Because of that, we have 2 layers of replicates. We have biological replicates and we have technical replicates. And so both are useful, but I will tell you kind of from the beginning that biological replication is the most important. So if you have a fixed number of runs that you can afford, you are better off having different biological samples in each run, as opposed to analyzing 1 sample really, really well with many technical replicates, but just having only 1 biological subject. And we will talk more about this in a minute, right? But biological replication is the most important. Okay, any comments on that? All right, so a fundamental principle 2 is randomization. So again, let's think about a very simple experiment where we compare a peak for healthy subjects and a peak for the disease subjects. So we want to compare 2 groups, healthy and disease. So let's just kind of assume, for the sake of the argument, that the healthy subjects were all acquired in day 1 and day 2 of the study, And then all the disease subjects were all acquired in day 3 and day 4 of the study. So there is a difference between the groups, which may as well look systematic, but now we have a confounding, because the difference between the groups can be due to 2 sources. It can be due to the disease, but it can also be due to the fact that the last 2 days had something special about them which changed the peak intensities. Maybe it was humidity, maybe it was temperature, maybe it was batch materials, maybe operator. All kinds of things can happen such that the peak intensities in the last 2 days changed more systematically, or are somewhat different from the first 2 days. And I think you probably will see examples of that with real data as well this week. So this is an example of a lost experiment where you cannot make any statements regarding the disease because the changes may be just as well due to the problem with the experimental protocol. And this is the case where no amount of statistical analysis will save that. So this is a really important part. And you know, in my experience, the changes due to the time of acquisition are usually way larger than any biological signal. So if you have that, it's pretty much the lost cause. So now this is what happens when we randomize. So when we randomize, we randomly allocate the biological samples to different days. And so now the 4 days are kind of randomly spread in the healthy subjects. They're also randomly spread around the disease subjects. So clearly we have more variation now because the variation around the mean now has contributions from the natural biological variation, but also day-to-day variation. So the fact that we randomized and we have spread kind of the healthy subjects over more days, well, inflated the variation, right? Because now we also have the technical day-to-day variation, but at least we have no bias. So now it's not an efficient experiment because it has a lot of variation, but at least it does not have a bias. And so especially in large studies, when we randomize all this kind of type of possible things which can affect the peak intensities, they will be roughly spread out equally between the groups. And so randomization in particular is useful when we know that there are some sources of variation which can affect peak intensities, but we do not know what they are. So then randomization is pretty much the only thing that we can do. And we never know everything which can affect peak intensities. So randomization especially protects us against the sources of variation which are unknown and we cannot control. And again, because we have 2 types of replicate, biological and technical replicates, we have 2 types of randomization. So random selection of subjects from the population to account for the biological variation, and randomly allocate samples to all the processing steps. So randomly select the samples from the population and randomly allocate samples to all the processing. And maybe I can also comment here that by random, I do not mean just kind of close the eyes and pick somebody, right? Because humans are really bad at doing that. Humans have very bad concepts, kind of just naturally, about what it means to be random. And so when you would like to do that, so the best way to proceed is generate random numbers in Excel or in R or in any other software. Fix that, and then follow that exact protocol. So that's really important, okay? Because don't trust yourself that you think, okay, let me just shuffle something. That's never gonna work, okay? So that's the important part, right? So randomization is really key. So replication randomization, it's simple, and it can really help us avoid a lot of trouble already. So here's an example. I really like this paper because these are statisticians. They used to be at MD Anderson Cancer Center in Texas, and they do a lot of consulting and collaborative work, and they're very influential in the kind of area of reproducible research, correct statistical analysis. And so in 2005, they wrote a paper which, unlike many other papers, which talks about successes, so this paper actually talked about failures. So they were courageous enough to compile a collection of failed studies that they had to be involved in, just to give examples of things which went wrong in real life. So, well, that's an old paper, right? So it sold it off, but it really doesn't matter. So this was serum samples with 5 types of cancer, and they had these samples anonymized, meaning that the statisticians did not know which samples came from which type of cancer. They did signal processing, normalized, peak, they did all the right things, and they did a class discovery experiment where they said, okay, I don't know what are the labels of the samples, let me just do some kind of clustering here and look for samples which are similar in the space of proteins. And so they plotted that, and so they saw that instead of 5 groups, they had 6 groups or something like that, and they were very excited. They thought, well, maybe we have some new subtype of cancer which have not been seen before. But so what they did after that, they looked at the same IDs, so they plotted the same IDs of the samples here that they had here, but on the y-axis they plotted the day when each sample was acquired. And so you see that the samples cluster perfectly by the day of acquisition. So what I was saying before actually really holds, that the day of spectral acquisition plays a really important role in in quantification. Now the problem is that the first several days were some very specific cancer subtypes which were acquired first, and then you had some other cancer types which were acquired last. So this is an example of confounding again, where the experiment is lost because they cannot claim any kind of patterns related to the disease because all the patterns are confounded with the day of acquisition. So this is an example of wasted money and time.
They would do something like look at something in the data set that they know is not changing time and possibly figure out
It's very hard, because all these normalization approaches, they're based on the assumptions, right? So you would, for example, you would say, I have some kind of standard. So now I assume that whatever day effect happened affect every single protein the same way as affected standard, right? What do we know? So then, our results rely a lot more on our assumptions than on the actual experimental evidence, right? And so it's just as good as the assumptions at this point, right? So you may have assumed this without doing any analysis, right? So usually, if you really want to provide strong experimental evidence, you'd better not make assumptions like that, right? So probably they could try to do something, but it would not substitute now having to go back and actually redoing it, right?
So when I talk to people who put the samples, the counter argument is, well, then I need to do less washing because then I have only carryover within my healthy and carryover within my disease. So I group all the healthy together, all the disease together, and then I don't have carryover from healthy to disease, and I have less washes. So it's faster.
Well, the goal, though, is not to conduct a perfect experiment which has no carryover, right? The goal is to learn about the biological variation in the actual disease, right? And so, yeah, well, you did a perfect experiment which had no carryover, but you introduced additional variation which now confounded your biological variation up front, right? So now, the third fundamental principle is the one where you can actually take into account better in a more efficient way some things which you know about, right? So carryover, it's kind of tricky, right? But if you have some additional things, like for example, you need to change a column in the middle of the experiment, or change some kind of material, right, or reagents in the middle, or you have a new operator coming in because the experiment runs too long, you can actually do things about that, okay? But as far as the carryover, you can work on minimizing this on the experimental side, right? But it's really not worthwhile to introduce bias due to all the other things which we know exist, right? Just to minimize the carryover. So, yeah. So what you would do in this case, so what is possible, well, let me talk about this, and then I will come back to you, okay? Okay, so let me tell you about the third fundamental principle, and then it will be easier to explain what I mean. So blocking is the third, and this is the most advanced principle of experimental design, which is a little bit more difficult, but it is very effective in that it helps reduce both bias and inefficiency. So let's take a look at the example again where we had a day effect, right? So if we have healthy and disease, so if we randomize between healthy and disease, so there are two potential problems. So one is we inflated the variation because now the variation has the day-to-day effect in addition to the biological variation. And the other is, well, if we only have 4 replicates and we completely randomly assign every sample to every day, it is still possible that somehow we have the healthy which were assigned to earlier days and disease somewhat to later days, just due to the kind of randomization artifacts. So in small sample size, it's still possible, right? So what blocking would do, so blocking imposes a restriction on randomization. And it says, I don't want to randomize everything, I want to randomize while taking into account sources of variation that I know about. And so in this case, if we know that we have a day effect, which really affects peak intensities, what we can do is to systematically allocate 1 sample from healthy and 1 from disease on day 1, then 1 other healthy and 1 disease on day 2, on day 3, and day 4. So it's important that every day the samples are selected randomly, right? So we select randomly 1 healthy and 1 disease, but we have exactly 1 from each group on the first day. And we have exactly 1 from each group on the second day, and so on. So now what we can do is, well, first of all, we see, okay, we have a lot of day-to-day variation, clearly, right? But within 1 day, we see that we have systematic change that points to the same direction. So what we could do, we can look at the differences between healthy and disease on this day, healthy and disease on this day, on this and on this, and now study these differences. And in this case, they're all positive. They're always systematically higher than in disease than in healthy. And so this effectively allows us to eliminate the day effect, because now we study differences within the day as opposed to averages over all the days in every group. So instead of having 8 data points, we now only have 4 differences, but these 4 differences, they're much less noisy. And so in this case, it will probably be difficult to say if there's a difference between healthy and disease, and here it will be easier to say if there's a difference between healthy and disease. But see, this requires 2 things. 1, recognizing that we have a day effect and allocating the samples in this smart way, right? And then 2, analyze the data with the understanding that we had this blocking, and we now analyze differences as opposed to comparing the averages. So this is why it's so important to talk to statisticians before designing the experiment, because we can talk through all of these things. We can discuss what are the important sources of variation which can occur. And we talk about how to block and how to allocate our resources in an efficient way with the analysis in mind. So you can only do this type of blocking when you know how you will be analyzing the data later. And so this is why you really need to have the whole understanding of the procedure already at the design stage. Because even if you blocked, but you didn't recognize at the analysis stage that you had the blocking, you will still be comparing this mean to this mean, and you will be losing the advantage of this design just because you didn't know that there was a blocking in there. So that's the important part. So again, blocking is a restriction on randomization where we randomize here, right? But we fix the number of measurements within the day. Now going back to this carryover effect, so what you can do is to block during the time. So for example, you have pairs, healthy and disease, and so you randomly allocate, do you have healthy first or disease first, this group. Then you have the next group, healthy and disease, shuffle. Next group, healthy and disease. So clearly you'll have this carryover effect, right? But because you kind of block pairs from each group, you will be able to have more similarity, right? So you don't have the accumulation of carryover through the entire experiment. You have pairs of healthy and disease kind of close in time. And so this is something that is typically done when you kind of worry about the time effect, which carryover is essentially time effect as well. Does it make sense? So you cannot avoid. That's right. So what? So you have to. So that's unfortunately part of the variation, right? So you have healthy and disease first, but then the next one will be disease healthy, right? And maybe again healthy and disease. So you shuffle the order within each pair every time, right? But at least you have pair of healthy and disease at different time points, as opposed to all healthy first, right, and all the disease after that. I think there was it.
What would be the most common statistical tool to analyze with, like ANOVA testing, or
Wait, so we're not talking about the analysis yet, right? But so
Okay.
yeah, exactly. So linear models and ANOVA, that's what you would, for sure, right? So that would be clearly the type of models that would be very helpful, indeed. And so in fact, when you design the experiment, so you think about the analysis, and so here, see, I'm talking about the differences, right? And then the differences of differences, clearly, this is what analysis of variance and linear models would do. But it's not the only approach, right? There are many other approaches as well. In particular, it depends on what are the measurements. So for targeted experiments, if you do log transformation, many of the assumptions of the ANOVA will be verified. But if you did, for example, spectral counting, you would have had to do something else because you have count data. So there are additional considerations. But for our discussion here, since we talk about targeted methods, right, so ANOVA is a very good way to think about that.
Regarding randomization, when you have groups within groups, let's say fractions within the disease and the healthy population, do you have a particular randomization strategy for that?
That doesn't matter as much because, well, you have different fractions. You would not be comparing this fraction to this fraction, right? You will take this fraction, compare across the groups, right? And this fraction across the group. So you can do both ways because what's important is that your comparison across the groups does not have a bias, right? So you could potentially analyze the first fraction completely and then the second fraction, or you can kind of interleave them. So that's the part where carryover can kind of be optimized a little bit because you're really not comparing between fractions, you're comparing within, I mean, the same fraction across multiple samples, right? So that part you can kind of do both ways, depending on what's convenient.
Thanks.
Okay, so let me give you a little bit more examples on that. So this is again from the same paper. So another example like that. So this time they have two types of cancer. Again, cell-death off, normalized, peak, all the right things, right? So they did again the dendrogram. They found two groups, and they were very excited because they had two groups of cancer. What they saw right away is that these two groups corresponded to a change of the protocol in the middle of the experiment. And again, because this was more of one disease group, this was more of another disease group, they could not use this data in any way. However, protocol change, this is something which can be planned for, right? So as I was saying earlier, so if we know that the experiment just has to run for too long and you have to introduce some kind of changes in the middle, so a blocking would be a tool for that. So now what you could do is to allocate the same number of healthy and disease before change of the protocol and the same number of healthy and disease after the change of protocol. And then you will be looking at these differences and you will be looking at these differences and then kind of combine the differences across two stages of the experiment. So in this case, there won't be bias. But if all the healthy happened here and all the disease happened here, then this is a lost experiment. So again, if there are things that we know will affect peak intensities and we know that they will occur, there is a way to work with that. But oftentimes, experiments are complicated. So you have many fractions, but maybe you don't just have healthy and disease. Maybe you have a time course experiment. Maybe you have some kind of time points for disease subjects. Maybe you have multiple treatments, not just healthy and disease, but also some kind of additional things. So very quickly, these things can become complicated enough that it's not so simple. And so this is where statisticians can help, where we can kind of look at all of the things which contribute to the variation and help allocate the resources which account for all the groups and time points and dependencies between the data, and also all these technical artifacts. But for simple things, when you have, say, two groups, right, it's pretty simple to do it kind of without additional kind of complications. Okay, so because we have two sources of variation, right? So biological and technical variation, we also block in two different ways. And so here, there's a slightly different vocabulary sometimes. So when we block with respect to biological risk factors, it's actually referred as matching. So here, if we have healthy and disease population, so we say that they're similar as far as all the risk factors, but they're quite heterogeneous still, right? So we can have male, female, young and old, you know, different weight, different smoking, and so on. So especially in studies of small sample size, it is easy, even if we randomly sample subjects, it is easy just by random chance end up with all the male and kind of older in the healthy group and then all the younger females in the disease group, even though we kind of thought about this and randomized just because the study is small. If the sample size is large, this will all average out, right? But for small sample size, it is actually quite possible that it will happen. So matching will again impose a restriction on randomization, and it will say, okay, I want to make sure I have young females in each group. Now I want to have older females in each group, and maybe I want to have younger males. So you can stratify populations by risk factors that matter, in the sense that they can affect protein abundance, and then we restrict so impose the condition that each group has the same number of subjects from each of the groups. So here's an example that we had long time ago in one of the studies. So it was a study in collaboration with Munich Heart Center, where for every incoming patient who had a consent, they would take blood samples and just put them in the freezer for future analysis. And so their biobank looked like that. So they had 5 disease groups. So age and gender were viewed as kind of important confounding factors. So our collaborators stratified this by 2 groups of age and gender. And so these were the number of samples in their kind of possession. And so clearly there are very different numbers depending on the groups. And they wanted to select a small number in the study. And so what you need to do then is to make sure that for the same combination of risk factors, you have the same number of subjects across disease groups. Now, each line doesn't have to be equal, right? You can have different numbers for different lines, but the important thing is that the numbers will be the same across disease groups. And so, for example, here, clearly this third line is limited to 5 subjects because these 2 groups only had 5 subjects, right? And then others could have more, right? So this would be an example of matching where we restrict the randomization to make sure that each disease group has the same number of biological replicates with the same confounding factors. So another example of blocking is when we block with respect to some technical sources of variation. And so a very specific example of blocking is multiplexing. And so there are many ways to multiplex, but I guess for our discussion here, an important example is when we spike labeled reference peptides in the biological sample prior to mass analysis. So this would be one example like that. So we have healthy and disease. So we add heavy labeled peptides, either synthetic peptides or maybe with some other type of labeling in the sample. And so now within each run, we don't have one peak, but actually a pair of peaks, right? So this is the endogenous healthy, and then this is the reference one. And this is, again, endogenous disease, and this is a reference one. And so now we have a blocking because within each run, we have a pair of peaks which go together. And the important part is that the labeled references do not have the biological variation, so they're as constant as possible. And then the endogenous peaks have the biological variation of interest. So here's a cartoon that represents that. So we have healthy and disease. So the horizontal lines are the means of each group. So if we just want to compare healthy and disease, we see that there's a lot of variation between the groups. So it's really hard to say if the change between the groups is systematic or not. But now the black dots, they're the corresponding reference intensities. And so remember, the reference intensities, they're supposed to be constant. But we see that we have a lot of variation between the reference intensities in this cartoon. So this means that we just happen to have a lot of technical run-to-run variation when we acquire the data. And so, because all of these black points, all of these difference in intensity are not due to biology, right? They're just due to some kind of technical artifacts. But again, just like we did with the day effect, what we can do is to look at the deviations of each of the endogenous peak from the corresponding reference. So this is for healthy, and this is for disease. And again, this is on the log scale, right? So the difference of log intensities is log ratio, right, between the endogenous peak and the reference peak. And so what we see here is that the deviations from the references for the disease are systematically larger than deviations from the reference of the endogenous. And so again, what we can do, we can compare these differences to these differences, and it's much easier to see that they're larger. right? So this is, again, an example where multiplexing, so in this case, just adding reference standards in the samples, again, leads to a more efficient experiment. But this will only be useful when we have a lot of run-to-run variation. If we don't have much run-to-run variation, then all of those black dots will have the same intensities, roughly, and then it's not really necessary, right? We could just as well compare directly the blue dots to the red dots. So if we have a lot of technical variation, then references are useful. If we have an experiment such that the run-to-run variation is very small, then we're wasting our resources because we have to pay for labeled peptides, and then we waste half of the transition space to measure references that we're not interested in. So that's kind of also the consideration, right? And usually you use some historical data or pilot studies to see if it's actually useful to have the references added. So this is really kind of the important point, right? That usually people say, well, you know, somebody used to say, I don't like to work with statisticians because they ask for replicates, right? So the thought is that replicates are the only way to increase some kind of the efficiency of the experiment. Clearly, you need replicates to be able to understand how much variation we have. It's extremely important to have replicates, so don't get me wrong, it's true, right? But another role of statisticians is to help increase the efficiency of the experiment given fixed experimental resources. And this is the type of consideration, so blocking, so matching and multiplexing, which can help us get more of the data even given a fixed set of resources. And so this is something not to be underestimated, because this is a really important aspect. So we can get more of our data if we think through the analysis. But on the other hand, if this is too complicated, just replicate and randomize. These are the first two steps which can really will keep us out of trouble at the very basic level. Okay, and so this is just to kind of get back to that. So this is what we kind of try to do to minimize the bias, right, and minimize the inefficiency. So how can we now, just again to give you some sense of what's going on kind of in the mind of a statistician when we talk about this. So, okay, let's say we have a choice, right? We have a choice of having more biological replicates versus more technical replicates. How do we know that biological replicates are more important? So I hope it's kind of intuitive, but actually statisticians formalize this in a very in a fairly rigorous mathematical way. And again, I promise not to do much math, so I will just kind of give you a preview here. So what we will do, and well, it was a really good kind of comment on ANOVA, so you can use ANOVA type of approaches where you essentially look at the observed intensity of the features and you describe all the sources of variation which can affect the intensities. So we have the systematic signal between the groups. We have the random deviation from the group mean due to the biological replication. So in this study, we actually had multiple sample preparation. It was an old study where we did glycocapture, so we did multiple glycocapture kind of extractions from the same biological sample. And then we had multiple technical mass spectrometry runs from each instance of glycocapture. So here would be an experiment like that, where we can decompose the variation in the observed peaks into the contributions of all of these sources. And then, so remember, I was saying that we talk about the variance of this, right? Just to talk about the efficiency. Turns out that if we write it like this, we can actually theoretically describe what the variance of our difference in means would be. And it looks like that. Okay, so here we have the biological variation, sample prep variation, measurement variation. The important part is here. So this will be the number of replicates. So K is the number of technical mass spectrometry replicates, right? So mass spectrometry runs. So if we increase the number of mass spec runs from the same biological sample, we increase K. So it means that this part of the variance goes down, but it does not affect any of this. So if now we do more sample preps, so it will reduce these two parts of variance, but it will not reduce this variation here. So the only way to dramatically reduce the uncertainty associated with the difference between the groups is actually to increase number of biological replicates, because it then affects all the components of the variance. So this is why this is how we show technically that biological replication is the most important, because it will be the most effective in terms of the reducing the uncertainty associated with the signal in the data. And the same thing actually with the blocking, so this will be kind of similar. So we have the systematic signal between the groups. Here we have the random deviation due to the block, for example, day-to-day variation that we talked about before. This will be the biological variation, this will be the deviation due to the measurement error. And again, we can write out these variances. If we don't block, then we have this day-to-day variation which contributes to the uncertainty. But if we block, we essentially got rid of the day-to-day variation when we look at the uncertainty between the groups. And so this allows us to kind of using, for example, a pilot data set when we can quantify the extent of variance components, we can actually decide here, is blocking useful or not. So for example, if this day-to-day variance is small, blocking is an extra trouble, right? So probably it will not reduce our uncertainty a whole lot. On the other hand, if the day-to-day variation is large, then it will be really an important reduction in the variance between the groups. And so using some pilot data sets, we can actually quantify the benefit of blocking before we even start the experiment. And that's really how it works kind of formally on the statistics side. Any comments on that? Okay, so one more aspect of design which people often ask about is pooling, and so I wanted to also mention that. Pooling means that we combine biological material from multiple samples into one prior to mass analysis, right? And so usually it is done for multiple reasons. For example, if we work with some very rare samples where we don't have enough biological material from one sample, we can combine that. But sometimes, we just have too many samples, we have few runs, so we'll just mix together multiple subjects and then we would profile that. So is it a good idea or not? So one thing which is a really, really bad idea is to do this. Take all the healthy subjects, mix them together, take all the disease subjects, mix them together, and then we'll have one healthy run and one disease run. Because this is exactly the situation which introduces the bias, because now we don't know if the difference between the two samples is because it's healthy and disease, or is it because this was acquired first and this was acquired second. Plus, we don't even know what is the variability associated with each group, because we only have one number. So we lost all the replication, right? And we introduced the bias. So one thing to never do is to mix all the healthy together and all the disease together. Now, this said, what we can do is do something intermediate. For example, let's say we have 10 healthy and 10 disease. Well, we can mix 2 healthy, and then 2 other healthy, 2 other healthy, and 2 other healthy, right? And then do the same, 2 disease, 2 disease, 2 disease. So now we reduced the number of runs by half, right? But we still have multiple replicate pools in the healthy group, multiple replicate pools in the disease group. So this gives us the estimate of variation, right? And it allows us to randomize to avoid bias. So these are really important things. Now, just as before, I mentioned how statisticians evaluate different designs, so we can continue doing it here. And so this would be an example of variance when we have the biological variance of the individual subjects, maybe sample preparation, maybe error. Now, if we have pools of R subjects, so it reduces the variance. So if I is the number of runs, if each run actually is a pool of R subjects, it reduces the variance. So from the mathematical perspective, it is beneficial if we have fixed number of runs. Let's say we have 20 runs. So if we have 20 subjects, or if each of the 20 runs is actually a pool of multiple subjects. So we effectively have more biological replicates within the same number of runs. So this is beneficial because it reduces the variation. Now, there are several caveats here. So even though pooling is helpful, so there are multiple disadvantages. So pipetting errors, right? So the important kind of assumption is that every mixture has equal contribution from each sample. But if we make mistakes and the pool actually contains more of one sample than the other, then it kind of introduces potential problems. So if you have a contaminant sample or some kind of failed sample, it fails the whole pool, so you lose more biological material this way. And another important thing is that by mixing things together, you kind of average protein abundances between multiple biological samples, which means that your goals of analysis are now limited to comparing population averages. So you cannot do a pooled experiment and attempt to do predictive model where you do biomarker discovery, where you try to predict the disease status of the individual patient because you lost the individual patients. Right now you only have pools of multiple subjects. And then there are also some technical caveats because oftentimes we work on the log scale, but we mix samples on the original scale. So there are also some kind of caveats with these calculations, kind of technically speaking. But generally, it is a good idea, provided that it's done correctly. Okay, so this is for the design. So any comments on that? Okay, so let me start a little bit talking about data analysis, and so I will finish when we run out of time, and then I will continue again on Thursday with that. So let me just kind of walk you over a very simple approach, so essentially comparing two groups using a t-test. And I would like to do this because I would like to clarify some important concepts which sometimes get mixed up when we do this comparison. So then on Thursday I will talk about additional things. So let's go back to the simple example of LCMS, but it would be the same with SRM. So if we have proteins digested, so we have individual peaks. Let's say we have one peak per protein, just to make it simple. And so we have replicates, so biological replicates, as we discussed, right, for the healthy group, biological replicates for the disease group, and we want to know if there is a change between the groups that is more systematic than as expected by random chance. So usually the way we think about this is that we start by assuming status quo, or kind of the not interesting case, right? A worst case scenario, meaning that we have no difference between the groups. So we assume that these two means are actually the same in the underlying populations. So remember, let me kind of take you all the way back here. So we are using the observed data to make conclusions about the populations. So these constants that we have in the populations, they exist in nature somewhere, but we will never ever know what they are, right? Because these are averages over like thousands and millions of people, right? So they exist, but we don't know what they are. However, we can make guesses, right? We can make some kind of assumptions or guesses about what is going on with the population. And so the way statistical inference can proceed is, well, let's assume that there is no difference between the groups. Is the data consistent with that or not? If the data is consistent, well, then maybe there's no evidence that our assumption was incorrect. However, if the data is very different from our assumption, well, then it's evidence that our assumption was wrong. So that's how we proceed. So we'll make some kind of guesses or assumptions and then see if the data corroborate this assumption or not. So we start by a simple situation where we say, let's assume that in the population, there is no difference between the groups. Okay, so let's assume that in the underlying millions of subjects, on average, the proteins do not change in abundance between healthy and disease. And then the alternative would be, in fact, there is a change in abundance. So what we will do, we'll have calculate a quantity, which we call a test statistic, which essentially is a signal-to-noise ratio. So the signal here is the difference between the group means. Okay, so we'll have the mean of the log intensity for the healthy minus mean of the log intensity for the disease. And the important part is the noise, right? So the noise is the estimate of variation associated with this difference in means. Okay, so there are multiple things that go into this. So there is the estimate of variation associated with the mean of the first group, and estimation of variation associated with the second group. So let me maybe give you a little bit more details here. So S here will be the variance, empirical variance calculated for this group, and this will be the S2 will be the variance associated with this group. So N will be the number of replicates in the first group, and N2 will be the number of replicates in the second group. Okay, so this variance in the group divided by the number of replicates, this is the variance of the mean of the first group, right? And the same here, this is the variance of the mean of the second group. Now, if we take the square root of this, this will be the standard error. So it just takes us from the square kind of scale to the original scale. This is the measure of uncertainty associated with the individual mean. And so if we want to compare group 1 to group 2, so we compare we combine the variances of the individual means, and we take the square root of them. Okay, so this is the estimate of noise associated with the differences between the means. So this is very useful. So now what I would like to make sure that we kind of distinguish is what it means to talk about standard error versus what it means to talk about standard deviation. So S1 is the standard deviation here, right? And this quantity is the standard error. So standard deviation characterizes the variability in the population. Standard error characterizes the confidence in the mean of multiple replicates that we have from this particular group. And so it's a really important, right? So standard deviation expresses the spread of the replicates in the group. Standard error expresses how confident we are in the mean. And so clearly the larger the number of replicates, the smaller the standard error, right? Because we're becoming more and more confident in the mean of these replicates. So there are a couple of things which are important here. So one is we're talking about the mean of the log intensities. And so oftentimes, again, if we go back to ANOVA, so a lot of times we rely on assumptions of normal distribution. So things that look like that, where our replicates have a kind of concentrated around the mean and they kind of taper down in a bell shape, right? So we have relatively little likely data points in the tails, more likely data points around the mean of the distribution, right? So I'm kind of trying to express it here. So t-test would rely on the fact that the means are distributed according to the normal distribution. So here's an illustration of that. And by the way, this is a very nice series which I very much recommend. It's called Point of Significance in Nature Methods, which is a collection of very short articles, like 2-pagers, which talk about some very kind of specific targeted topics in statistics. And so some of these illustrations here are from this series. I really like it because it's a very short read. And so you look at these illustrations, but you can go back to the actual vignettes and read more about what I'm talking about here. So one of the things that people kind of ask about is, well, what if my peak intensities are not quite distributed according to normal distribution around the mean? Maybe I have outliers, maybe I have some kind of heavy tails, maybe I have some kind of things which misbehave. So the important point here is that we rely on the means being normally distributed and not necessarily the individual data points. So here's an example where we have different types of distributions, which can be all kinds of weird things, like this one is a really irregular distribution. And so what these authors did were they took, well, let's take 3 data points from this distribution, calculate the mean. Let's go back, take another 3 data points, calculate the mean, and so on. And so now we plot the histogram of those means of 3 data points taken from the normal distribution. Now here we calculate the histogram of 3 data points taken from this distribution. Here it's the histogram of the means of 3 data points taken from this, and so on. And so you see that as the sample size increases, even though the initial distributions look differently, eventually the distribution of the means become more and more bell-shaped and more centered around the mean. So this is what is called the central limit theorem. So as the sample size increases, even though we start from some kind of really irregular things, oftentimes the means themselves can actually be assumed to follow the normal distribution. And this is very helpful. So you see that clearly if we start from a nice case, these distributions are much more concentrated around the mean than if we start from some kind of bizarre case. And so it takes longer for irregular distributions to look like bell-shaped, but still, eventually, with a large sample size, normal distributions are often actually quite plausible. So this is one kind of caveat. But another caveat is that oftentimes we actually don't have this sample size. We have a fairly small sample size. And so with small sample size, we run into all manners of problems. And so this is another example from the same series which illustrates that. So they took this kind of irregular, bizarre probability distribution, which looks like that. So they have on the x-axis here is the sample size. So it starts from 10 replicates all the way to 100 replicates. And so here they calculate the mean of, well, that many data points taken from this distribution. And they repeat it 3 times. And so you see that when the sample size is small, the individual means can kind of go all over the place. So eventually they will really represent well the mean of the distribution, but for small sample size, there is quite a bit of variation. Well, so this is why statisticians ask for replicates, because we can see the mean much better if we have more replicates, right? If we have few replicates, it looks like that. So it may really be quite far away from the real mean of log intensities in the group. But the worst thing is here. So this is standard deviation, right? So remember, standard deviation expresses the spread of these distributions, right? So this is S1 here. So it expresses how broad this distribution is that we start from. And so if we have a small sample size, you see that it actually tends to underestimate the variability in the data. If it underestimates the variability, it looks a lot more optimistic than it really is. And why is that? Well, if you only have 3 replicates, so it's likely that it will come from some kind of region here, which is highlighted in the box. Only 3 replicates may not be able to sample this tail or this tail. And so now if you calculate standard deviation based on these points, you do not recognize that you have a much broader range possible, because you only had 3 data points here. And as the result, the variability looks less than it really is in real life. And so we underestimate the variation, and so we will end up with false positives, right? Because we think that the data are less variable than it really is. And of course, if we have more replicates, then eventually we'll start sampling the entire range of the distribution, so the standard deviations converge to the true values. But for the small sample size, this is one of the reasons why the results are often too optimistic. And of course, it affects the standard error. So as you see, the standard error goes to 0 as the sample size increases, but it's somewhat also variable in the beginning, right? So this is another point, just to reiterate, standard error goes to 0 as sample size increases, meaning that we have less and less uncertainty associated with the mean, but the standard deviation converges to some kind of constant, because it talks about the spread in the population and not in the mean. And so this is really important distinction, because oftentimes people are confused between these two, and so it is not clear what we expect, right? So standard deviation will stay constant, standard error will go to 0 as the sample size increases. So now let me just kind of, oh, actually I'm over time. So how about I stop here for now, and then on Thursday we will finish this and I will talk about more advanced methods for the analysis. Does it work? Okay? Unless you have questions, so maybe let's take a minute or 2 for questions.
Hello, thank you for the very nice presentation. I would like to ask you, in let's say in graphical representation, for error bars, we use standard deviation or standard error?
I have a whole series of slides on that.
Okay.
Let's hold on to this thought until Thursday, I guess. I just want to make sure all the other speakers are on time, so I don't want to go overboard. But, well, but the short answer is, you do what makes sense for the point you want to make, but make sure it is clear what you are plotting and what are the conclusions from that. Because the worst thing that I've seen that people just say, and we have error bars. And they just go like that. And it can mean all kinds of things, as I'm going to show you tomorrow, I guess. It can mean very different things depending on what these error bars mean. So they all are useful, right? But make sure you explain what they are, and then the conclusions have to match the things that are displayed, right? So.
Thank you.
Mm-hmm. Okay.
Maybe I'll ask a question, actually.
Oh, yeah, please.
So I, for example, don't work with disease and healthy. I usually work with cell lines. And then a question that always comes is, should I make one big experiment and let's say 3 plates from each condition in a time course? Should I grow this experiment 3 times? What would you recommend for people that do time courses in cell lines and not healthy and disease?
Yeah, you want to have 3 different plates and 3 different instances of growth. Yeah, but cell lines are nice because they're much more controlled, right? So you'll have a lot less biological variation than humans, obviously, right? But 3 different plates and just start from there. Yeah.
Any other questions?
Just a short question about normalization, because
Yeah.
you briefly touched on normalization. So you said you always take assumptions if you normalize. So what would be your suggestion for normalizations?
It's very specific to your experiment, right? And I will talk about this on Thursday, but maybe we can start. So usually you assume that something is not changing between runs, right? Because that you would want to normalize to, right? So if you are doing, let's say, DIA, right, where you measure many proteins, and your experiment is such that you think the majority of the proteins should not be changing, then it's probably fine to normalize with, let's say, median of the intensities across proteins, because you would assume that the median is stable, and so whatever changes are there, they will be somewhere kind of very rare, right, and scattered across the runs. On the other hand, if you're doing SRM and you have 50 targets, well, you selected them for a reason, right, because you think they're changing. So for SRM, this will not be an appropriate assumption, right, to say that the median of proteins is not changing. So usually if you have more measurements, and if you can assume that more are not changing, you can do that. If you have many things that change, you really need to rely on standards, right? So either reference peptides for each of the endogenous peptides or some kind of internal standards. But then you assume also that whatever affected the standard affected exactly the same protein in the same way, which is maybe also a big assumption. Plus, all these standards, you only normalize the artifacts which occurred after you spike the standards. So all the sample handling and all the upstream things will not be normalized, right? Because usually the references are added after that. So then there's another thing, you can try to use some kind of biological standards, right? Some proteins which you think are so tightly regulated that they're not changing. But I know that there's no such thing, right? It's very difficult to find a protein which you know, no matter what you do to your sample, it never changes, right? It's probably not possible, right? So it is very difficult. It is very difficult. So this is why kind of replication and randomization will kind of compensate to some extent for some of those technical differences, right? So if you have a large enough experiment, even if you have some of those artifacts present, sample size will compensate for that. If you randomize, at least this will not introduce the bias, right? And so usually our solution is kind of all of the above, right? So large enough sample size, randomization, normalize with respect to some standards, with the understanding that some of this will probably not be normalized away, right? And you just kind of proceed like that.
About the pooling, so I was wondering whether you can sort of quantify the benefit from this. So say you had 100 samples, but you can only shoot 20. So you can either choose to pool or you can choose to pick 20 out, right? So can you say in a sort of power analysis kind of way how this increases?
That's exactly what you would do, right? So what you would do is you would need a pilot study where you can have the numeric values for, let's say, biological variation and then technical variation. And then you can calculate theoretically the variance if you would have without pooling. You will have the variance with the pooling. And so the variance, that's what defines the sample, kind of the power and the number of replicates, right? And so now the result will depend on the interplay of how big you have biological variation versus technical variation, how big is I versus R, right? So this will be the number of samples in a pool, right? And so this is number of runs. And so you can kind of play with the different combinations of number of runs and number of samples per pool. And you would assume that your future variance components will be similar to the variance components in the pilot study, and you can kind of play with that.
So it's usually you know something about the technical variance, but often you don't know something about the biological variance up front, right? So can you make an assumption? Is it usually, can you make some assumptions on this side? And does it?
It's like always, right? So you're trying to predict the future, right? So the more specific you are about what to expect, the better your outcome would be, right? So if you assume something which is off the mark, then your results will be very different from what you assume. So either you have some historical data, and you don't need kind of a very large number of replicates, right? So you would take some kind of limited number of biological replicates to estimate the extent of biological variation in your system, right? So on your samples, with your kind of sample prep and sample storage and your instrument, right? Then you can be much more kind of exact about what will happen. Or you say, well, I had a friend who had something else, right? He had this variance components. I do this calculation, but this may be very different from what you would have, right? So it's like always so the more data we have as opposed to assumptions, right, the better our result would be. And so if you would try to make the assumptions, then, well, how do you know that your assumptions are good ones, right? So you may not as well even do any experiment, right, to just assume things, right? But the goal is to actually have some kind of evidence that you can bring in to do a more educated guess about how to proceed.
I guess there's a question. I mean, is there a case where if you don't know the biological variance, that the pooling will be detrimental, so that the case where just shooting the individual samples would have been better?
Well, so that's kind of, these are the problems with that, right? So it depends a little bit on your goal. So for example, if your goal is predictive analysis, then you don't want to do pooling, right? So another thing what I mentioned technically is that these calculations of variance and power calculations assume that you average on the log scale, but what you actually do in reality, you mix on the original scale. So I'm not aware of any studies for mass spectrometry, but in gene expression microarrays, there is some literature which actually compares experimentally, so pooled samples and then the theoretical calculations. And so when the average on the log scale is similar to the biological averaging or not, and they found some genes for which it's really not, doesn't behave in practice as it's supposed to behave theoretically, but it was a small proportion of genes. Things like that. So what you can do if you don't have any idea at all about biological variation, you can do sensitivity analysis, right? So you say, you can kind of work backwards, right? And say, okay, I can afford that many runs. Let's say I can do that many pools. You don't know the variance components. Well, try a few numbers and see what will be the worst case scenario, right? So you say, well, my worst case kind of biological variation will be that much. So what will be the smallest fold change I can detect with that? And then you can kind of play with it and say, well, how likely am I to have this large biological variation? It's really hard at that point if you really have no idea, right, what the numbers are. But if you have at least some idea from some previous studies, you can say, well, my worst case scenario would be that I cannot see anything between a 3-fold change. Is it good enough? Right. And then you can kind of play with that.
So we'll have a second lecture. We would like now to invite you for a coffee break, and then the next lecture will be given in the classroom upstairs at 11:15. Thank you, Olga.
Thank you.